Welcome to The Explainer. Today we're exploring the incredible, and let's be honest, sometimes flawed, architectural marvel that is the human brain. We're going to look at the actual blueprint for its construction and see what happens when the plans go just a little bit wrong.
Okay, so let's jump right in. You might think a baby's brain is just a tiny version of an adult's, right? But check this out. On an MRI scan, they literally look like photographic negatives of each other. Wild. The reason for this crazy difference is that a baby's brain isn't just small, it's an active construction site. And the key material being laid down is called myelin.
So, myelination. Think of it as the process of insulating all the brain's wiring. Here's the key. Immature myelin is mostly water, but mature myelin is fatty. Now, on an MRI, fat and water light up in completely opposite ways. So this fundamental switch from watery to fatty is what flips the entire image. It's why radiologists use different types of scans, a T1 for the first year and a T2 for the second, just to keep track of this crucial construction phase.
So, if we're sticking with this whole construction analogy, what happens when there are typos in the blueprint? Well, we can actually understand these congenital malformations by grouping them into three main types of construction failures. We're going to break them down into three distinct acts.
Let's kick things off with Act 1, failure to form. This is exactly what it sounds like. It's when a fundamental piece of the brain, a structure that should be in the original plans, just isn't built. So that brings up a really interesting question. What if the brain's main information superhighway, the thing connecting the left and right sides, was just missing? Well, that leads us to a classic example, a condition involving the corpus callosum. You see, without that corpus callosum bridge, the nerve fibers that are supposed to cross from one side to the other literally have nowhere to go, so they just pile up along the center. These pile-ups are called Probs bundles, and they physically push the brain's fluid-filled chambers, the ventricles, apart. This creates these really distinctive shapes that radiologists can spot, like steer horns from the front or a racing car from above.
Okay, now for Act 2, failure to divide. The error in the blueprint here isn't about missing parts. No, this time it's about structures that were formed just fine, but then they failed to separate or cleave from each other as they were supposed to.
This is a condition called holoprosencephaly. See, the brain is supposed to start as one single structure and then split into two hemispheres. And this process happens from the back to the front. When that split is incomplete, you get a whole spectrum of disorders where, and this is fascinating, the face can actually predict what's going on in the brain.
And this table really lays out that spectrum perfectly. On the mildest end, you've got the lobar form, where only the very front of the brain is fused. But as you slide over to the severe alobar form, there's no division at all. The brain is basically a single cup-shaped structure. And you can see how, as the fusion gets more complete, the outcome changes so dramatically, from being survivable to, tragically, just not being compatible with life.
Now we come to our third and, honestly, most dramatic stage of construction, the great neuronal migration. This is an absolutely epic journey where millions of brand new neurons have to travel from the deep, central part of the brain all the way out to the surface to form the cortex.
It is a seriously perilous journey, and the source material for this explainer uses a fantastic analogy from the Batman movie The Dark Knight Rises to describe it. It's like an impossible climb out of a deep pit, a desperate chant to rise up to the surface. This so-called climb has three main stages. First, the neurons are born. That's proliferation. Second, they migrate, climbing past earlier waves of neurons to reach the surface. And finally, they organize themselves into these complex layers and columns that create all the brain's characteristic folds and wrinkles. An error at any one of these steps, well, it can be catastrophic.
So what happens if the neurons don't make it? If they fail their climb? Well, if they don't reach their destination, the brain's surface can't organize and fold properly. The result is a condition called lissencephaly, which literally means smooth brain. This failure to fold can give the brain this really distinctive figure 8 or hourglass shape because the big major fissures are there, but all the complex smaller folds are just gone.
Another organizational error is schizencephaly, or split brain. This is where actual clefts form, running the full thickness of one hemisphere. And these aren't just empty spaces, they're gaps lined with misplaced gray matter, the brain's processing centers. Having your processing tissue in the wrong place is a huge problem, and it's thought to be caused by some kind of disruption during development, maybe like an injury to the brain's blood supply in the womb.
And this slide just illustrates the two main types so well. The more common one, open lip, is a wide fluid-filled channel that's pretty obvious on a scan.
The closed-lip form is a lot more subtle, because the edges of the cleft are actually fused together. But the telltale sign is still there, a tract of gray matter running right where it shouldn't be.
So, the crucial point in all this is that studying these blueprint errors doesn't just show us what can go wrong, it gives us this really profound, humbling appreciation for the near miraculous sequence of events that has to go exactly right every single time to build a healthy brain.
So let's just pull this all together for a second. We've seen that a baby's brain is a full-blown construction site, completely transforming itself in the first 2 years. And we've learned that these major malformations are basically blueprint errors that happen in one of 3 ways: parts that fail to form, parts that fail to divide, or the workers, the neurons, that fail to get to their final job site. And that really leaves us with a final thought to chew on. We're all walking around with this absolute masterpiece of biological engineering in our heads, and yet, its creation depends on this developmental dance of such insane complexity and fragility that its successful completion is, in itself, an everyday miracle.